Alright, thank you so much guys for coming. We're already reviewing midterm 3 for midterm 3, lectures 11 to 15. So the next time you come, there will be the exam. And as usual, before we go into lecture 11, neurons and glial cells, we talk about the format, which you are now used to. Is going to be the same as before, right? So we've got our section 1. So that's the usual multiple choice. And I'll probably set the quiz maybe tomorrow or so. On Connect, you have to study anyway before you do the quiz. So, if I finish selecting the questions, I'm going to do it today. So the usual 30 times 1, which is 30 points. Section 2 will be short answer questions, sometimes definitions, 5 of 6 times 2, 10 points. Then the final section 3 is long answer. 4, 5 times 5, which is equal to 20, 30, 60 points. Alright? Okay, so we'll start off with neurons and glial cells. Alright, stop. As we normally do with any system, we talk about the functions. So we know that the nervous system does what? It receives sensory information from the rest of the body, right? By sensory neurons. And then once it gets that information, it's going to be passed from one area of the brain to the next, and integration takes place, which is pretty much making sense of that stimulus and then generating a specific response, which falls under the control, right? So, control of muscles, for instance, for them to contract, glands, for them to release, certain glands, and all that is meant to maintain a constant internal or stable internal environment, I should say. All right. All those control mechanisms are meant to maintain, the best optimum conditions for the body cells and so on. Right. And then, of course, mental activity, which is thoughts, decision making, memory, all those kinds of things. All those are functions of the nervous system. And if we talk about them in the exam, as usual, when you say sensory information, you tell me a little bit about it, so that to show you to understand that function and what it means for the body, right? So the part of it, you've got to know basically how it's all organized, right? So the central nervous system is made up of what and what? Yeah. Right? And then the peripheral nervous system is made up of what and what? Huh? Made up of the peripheral nervous system. Spinal nerves and ganglion. Okay, because if I ask about the organization of the nervous system, guys, I want you to tell me what each of those sections or divisions includes. What are the parts that make up those divisions? Okay? And the sensory, that's one that is all those sensory nerves carrying information to the brain, either about the external or the internal environment, right? Okay, and it's not divided, but it's the motor division that is divided into two parts. One, the somatic, that deals with the muscles, skeletal muscles specifically, okay? And then the autonomic nervous system, which deals smooth muscle, really most part, viscera, cardiac, muscle cells, anything that's automatically controlled is dealt with by the autonomic nervous system. Okay, so this is skeletal muscle, specifically skeletal muscle only. All right, and then this one is smooth muscle plus cardiac muscles, Okay, smooth muscle is mostly in the viscera, like the small intestine, the digestive system, and blood vessels. All that automatically controlled. So we talked about it last time. The parasympathetic, what does it do mostly? What does the parasympathetic do mostly? It inhibits or stimulates? Huh? Yep, it inhibits. Okay? Remember how you tell them apart? Sympathetic, of course, will do the stimulation. If you're sympathetic to somebody, you want to give them something, you want to stimulate them, okay? So that's how you can tell parasympathetic and sympathetic apart, okay? So this one will stimulate. Okay? And then what did we say the enteric division or subdivision of autonomic nervous system does? What does it do? Enteric. Yeah, deals with digestive functions. Okay? This is a good 5-point question bonus. Just describing the organization of the nervous system. Alright? And how it's made up. So, of course, we need to know about the cells of the nervous system, and the major of which is the neurons. Okay? And in neurons, there's different types. Okay? Characteristics of neurons. What about them? Right? What do they have? They have, for one, they don't divide, right? Right? Because they are missing what? Centrioles. Very good. So already I'm talking about the characteristics of neurons. Right? And what else do they have? Because they don't divide and they don't have centrioles, they have a lot of this particular organelle. In fact, there's a special name. Well, the soma is the cell body. Meso body. Yeah. Meso substance. What is meso substance? What is that organelle, really? What is the meso substance? Made up of endoplasmic reticulum, right? And what's the function of the endoplasmic reticulum? Protein synthesis. So that if a dendrite falls off, you create proteins to repair. So that's why the neurons have a lot of repair organelles. You know, organelles that are meant to create new parts, spare parts, if you will. Okay, so meso body or meso substance. Okay, and this is really rough endoplasmic reticulum with a lot of ribosomes. Okay, for protein synthesis to repair. Okay, and of course they are excitable cells. Neurons are excitable cells, right? Because they can be excited and they can generate a neuron. Okay, and this is pretty much there's going to be many of these divisions. Okay, I'm only drawing a few because I don't have a lot of time. Okay, so this part here where the action potential originates, what's it called? The trigger zone or the axon hillock. Okay, and we call these short processes dendrites. Okay, those are all characteristics, and this is the cell body, like Brenda said, which is also called the soma, right? Okay, with the nucleus in there, and then this is called the axon. And then later on we'll talk about myelination. That's the only part of the neuron that gets myelinated. Okay? Alright. So functions, of course, they send and receive messages. Okay? This one, there's 3 types. The one I've drawn is what type is this one? The one I've drawn with many dendrites and 1 axon. Multipolar, yes. So this one is a multipolar. Okay? And then we get these ones. Okay, this one is a bipolar, right? Right? Okay, and then this one, of course, is the unipolar. Okay, what are the functions of them? Where did we say we find these mostly? Sometimes when I say, oh, can you tell me the 3 types of neurons and tell me what their function is? What they do, the characteristics sometimes. Motor neurons. All the motor neurons and the ones in this nervous system are all multipolar. In fact, the bulk of the neurons are multipolar. Alright? Okay, so these ones are motor neurons for the most part. And all the neurons in the brain. They are multipolar because they synapse with many other neurons or many other parts of the brain. It's all connected. Right? So what did you say bipolar and unipolar are reserved for? Yes, sensory. What kind of sensory information? What kind? Look at general senses, and these ones like eyes and smell is special senses. Okay, so these ones are really reserved for special senses. Okay, such as eyes, ears, smell. Okay, so I want you to be able to tell me all those things about, okay, the neurons. There's a lot you can talk about the neurons. Okay, all right. So good. Moving right along. So, and then the second cell type found in the nervous system, these are glial cells, or neuroglia for short, for long. Okay, that's the full name of it, is neuroglia. Okay, and we talked about it. There's 4 in the central nervous system, and there's 2 in the peripheral nervous system. Okay, astrocytes, we said they look like a. So that's a characteristic. Okay, and they've got many functions. So what are some of the functions of astrocytes? Okay. Not quite, but the insulation comes kind of here with the, but they anchor, okay, neurons, and they put them to proximity of nutrients, blood vessels, okay, and they have a big function to do with the blood-brain barrier that blocks all those, almost any substance really, except glucose, and neurotransmitters can go up there. Okay, what does it do? Because it's all over the synapses, it mops excess ions. Okay, because the business of neurons is to send ions in and out of the membrane to send all these electrical messages. So sometimes there's going to be some left around in the interstitial space, and the astrocytes will mop that up. Okay, so mop excess ions, and they're also involved in repair. Because sometimes I do ask you, can you please tell me about 5-6 functions of astrocytes, a specific glial cell? Usually I'm very specific about cells, and you have to talk about it. Okay, and you can give me a bunch of repair and also migration of young neurons when they grow up. When they are, growing, so that they grow and synapse with the correct organs. Okay, so they guide young neurons and so forth and so forth. So there's all sorts of functions that you can think about for astrocytes. Okay, now we talk about ependymal cells. This one's kind of, the name doesn't tell you what they do, but these ones, can anybody tell me what they do? Yeah, produce cerebrospinal fluid. Okay, what kind of cells are they? They are kind of sort of columnar with cilia on them. They're ciliated cells. And the cilia, what does the cilia do? Helps to circulate the cerebrospinal fluid. Okay? And they line special blood vessels called choroid plexus, okay, which is suspended inside of these ventricles, and it produces choroid plexus. CHOROID, okay? Okay, so that's what choroid plexus. These are specialized blood vessels that are lined up. Plexus is, that is lined up by these ependymal cells. So produce and circulate cerebrospinal fluid. How about microglia? What do they do? For starters, they are small. They're microglia. They're small cells. So few dendrite small cells. And what do? Function. Anybody? Immune cells, yes, these are macrophages. Yeah, thank you so much. Immune cells, macrophages, okay, they engulf bacteria and so forth, they protect the cells, of course. And now you can talk about the insulation. These ones are oligodendrocytes. Oligo means few dendrites, so also these ones are few dendrites. But their function is to produce, what's that? Myelin sheath, yes. Okay, which is what insulates. And the myelin sheath is found on the axon. But I think we'll talk about it later on. So produces the myelin sheath. Okay? Alright, so we're done with those ones. And then in the peripheral nervous system, the Schwann cells, what do Schwann cells do? okay, produce the myelin sheath in the periphery. Okay, and then satellite cells, what do? Satellite is support and nourish, and these surround ganglia. What is ganglia? Cell bodies outside of the CNS, right? So whenever a cell body is in the periphery, it's called ganglia. When cell bodies are in the brain and central nervous system, they're called nuclei. Okay, very good. So these ones surround ganglia. Okay, so ganglia are cell bodies, and then they kind of support and nourish them. Okay, cell bodies in the peripheral nervous system. So we talk about myelination, which is swan cells over there in the periphery and oligodendrocytes in the central nervous system, right? So this is like a fatty, whitish substance that gives rise to white matter and gray matter, right? Okay, so and it insulates. It insulates that part of the axon on which it is. All right. So that's one of the functions. It insulates that part of the axon, and then what else does it do? The myelin sheath, what's that? It speeds up the conduction of the action potential, okay? Why? Because the action potential is only going to be conducted in the spaces between the myelin sheath, which we call nodes of gravity, right? Okay, so speeds AP, action potential, AP, action potential. Okay, unmyelinated, let's say because I was thinking of AP. Okay, so functions and characteristics of those ones. These ones are not myelinated, okay, and they transmit slow because of that, okay. So these ones don't have the myelin sheath and transmit slow, but still they are surrounded by Schwann cells in the periphery and also surrounded by oligodendrocytes in the CNS. Why? Because they still need to be insulated. Even if they're transmitting slowly, they still gotta be insulated, okay? Because then otherwise they will cross-contaminate each other. I mean, not cross-contaminate, but cross-excite each other and not you get problems. Any electrical wire you see always is covered by a rubber. It has to be. Otherwise, it's gonna be live and it will hurt you. Okay, so very good. So gotta know the myelin sheath, what it does, the cells that produce it, okay, in the periphery, in the central nervous system. Okay, so because of myelination, we get white matter. White matter is made up of myelinated axons. If I were to ask you to define white matter, you'd say myelinated axons. Because only axons can be myelinated, nothing else. Okay, so this will be axons, myelinated axons. Okay, and then gray matter will be, what is gray matter? What is gray matter made up of? Anything that's not myelinated, which could be dendrites. Dendrites don't get myelinated, and yours, the soma doesn't get myelinated. Okay. What else doesn't get myelinated? Some axons. Okay, so some axons also. So if I was to ask you to talk about and describe gray matter, you'd have to mention all those things. Unmyelinated structures, which include some axons, you know, cell bodies and dendrites. White matter will be myelinated axons. Very good. Okay. We move on to action potentials. Now we've talked about the nervous system. Now we're going to talk about how it generates the messages, how it sends the messages along. And the way it does it is via those small electric impulses called action potentials. Right? So the electric signals that neurons use to communicate with muscles, other cells, glands, etc. Right? So, what is action potential? It's a large membrane potential change that is propagated along the axon or the length of the cell membrane. Okay? So these are large, membrane potential changes that reach threshold or go over threshold, and because of that, it is passed along the rest of the axonal membrane, all right? And then, of course, the functions are to send messages to other neurons or muscle cells, like in the neuromuscular junctions, all right? So how do we generate these action potentials? Okay, It's because of ion concentrations. Okay, because after all, what is an action potential? Movement of ions into and out of the cell. That's all it is. All right. And if we're going to be moving anything into and out of the cell, then we've got to talk about membrane permeability characteristics. Okay? So that's what's responsible for that. Because it depends on the ions that it is permeable to that gives it the ability to generate an action potential. Right? So if I ask about the source of the electrical signals, or how electrical signals are generated, then you've got to talk about, the permeability characteristics and ion concentrations. All right? Okay. Not only do we need these ions to be moving across and so forth, we need to generate a gradient, okay, to where we can separate the charges. We polarize the membrane so that we have got more positive charges somewhere and more negative charges on the inside, more positive charges on the outside. So we got to generate a gradient. And if I ask about how do we generate a gradient that is responsible for these action potentials, then you talk about the sodium-potassium pump, right? Because what does it do? It will pump 3 sodiums out, okay, and 2 K plus in, okay? That way you're beginning to have more positive on the outside, less on the inside. So on the inside it becomes more negative, okay? All right, and then of course we've got to have ion channels. The leak channels are not gated. They are always open, okay? And the membrane happens to have more potassium leak channels. Okay? So potassium tends to carry its positive charge outside of the cell. Also, which kind of creates a more negative charge on the inside of the cell. So that's how you create that gradient. Okay? So there's more K plus leak channels. Okay? And then of course we need gated ion channels. Okay? And for the most part, we use the voltage-gated ion channels. They are the channels that respond to a certain voltage, and then they open or close. Okay? That's what creates the gradient. So, know those 3 things that create the gradient and the source of the action potentials, which is the membranes, permeability, and the ion channels, or ion concentrations. Right? Okay, so moving on with electrical signals, we talked about it. We said there's 3 kinds of gated channel, and there's more than 3 kinds actually. Other, there's more here. Okay? So, like ungated, we know about it. The one we studied when we did the synapse or the neuromuscular junction, the one that binds the sodium like ungated channels. They bind to some ligand for them to open. Okay? So they bind to something, say acetylcholine, or any other neurotransmitters, serotonin, dopamine, whatever. All right? And then these ones respond to voltage to open. Like potassium channels open at plus 30 when it's peak maximum potential, and sodium's close. Okay? Sodium will open at plus minus 55 at threshold, and then a whole bunch of them open up and you depolarize. Okay? So they respond to voltage, and other ones could be mechanical. Sometimes it's touch, which is a mechanical. Okay? Sometimes it's physical deformation. You actually change the shape of the receptor, then it opens. Okay? So that's a physical one. So when it changes shape, that's physical. Sometimes it responds to temperature changes. Okay? Such as the receptor for temperature on your skin. Okay? And then an ion channel opens up. Okay? And you generate an action potential that goes to the brain. So there's many other gated channels, but for the most part we concentrate on voltage gated. Okay, so the action potentials. Alright? So resting membrane potential. Okay, what the definition is the membrane potential when the cell is not being stimulated, when it's doing nothing, when it's resting. Okay? And it is so cell doing nothing. Okay? You say it better in the exam, of course, okay? So characteristics of resting membrane potential. It's minus 70, right? Millivolts. If you start with that, then you know, of course, that the inside is more negative in, more positive outside. Okay, what else? You know, there's negatively charged ions. There's a table that gives you all the characteristics for resting membrane potential. All those big negatively charged ions that do not have channels cannot exit the cell, so therefore the inside becomes more negative. Okay? And then again, like I said, there's more potassium leak channels, so potassium tends to leak outside, again making the inside more negative. Alright? All those things. There's more things you can talk about the resting membrane potential. You really have to know resting membrane potential. Why? Because you have to know where you are in order to generate an action potential. You need to know where you're starting, okay? And then you know how to generate an action potential. Because all an action potential is replacing the resting potential, okay? So when I know those characteristics, sometimes in the exam I say, can you please tell me about the characteristics of the resting membrane potential, or you know, we talk a lot of times. You can come up with 8 points or something. Okay. There's only 2 ways by which you can change the membrane potential. Okay, resting membrane potential. Okay. So this is the cell at rest. Boom, boom, boom. -70, right? -70 millivolts. It's resting. It's resting. You can either start to depolarize, right? Create like a graded potential, which we say kind of sort of goes in decremental fashion, if it is at threshold. Or you can hyperpolarize. And then again in decremental fashion, it goes back to resting. Okay? So there's only 2 ways that you can do. You can change the membrane potential. You can either depolarize going up, or you can hyperpolarize going even further from threshold. Okay, so those are the only 2 ways. And to depolarize, what has to happen? You open sodium gates, and where does sodium go? What's that? Where does sodium go when you open sodium gates? Remember the conditions at rest I was talking about a while ago. What did I say? There's a bunch of sodium. Where? Inside or outside? Outside. So you have to know the conditions at rest, right? Like I said. If you know the conditions at rest, the rest you don't even have to memorize it. You can work it out. Because you know if it's going up, it must mean the inside is becoming more positive, right? You're going up, up, up. If you're going down, it must mean the inside is becoming more negative. And you can move any one of those ions based on where it is to create that positivity or the negativity, right? So you don't have to memorize that. You just have to remember all the conditions at rest. So Na will move into the cell because there's more Na outside. Okay? So hyperpolarization, well, you know, what happens here? You can open 2 gates actually for hyperpolarization. If you open potassium gates, potassium is more inside, so potassium goes outside. And it carries, its positive charge, which means it becomes more negative inside. Okay? Or you can open chloride channels, right? Chloride channels carries, chloride ions carry a negative charge. And where are they found? In or outside the cell? Outside. Because it follows sodium. Right? Sodium chloride, remember salt? So they love each other, so they always stay together. So wherever you find sodium, you're going to find chloride. So if you open chloride channels, it's going to move in. Everything always moves down its concentration gradient when you open the gate. Okay? So if you know the conditions at rest, if I tell you this gate opens, you know what it's going to do. And you know how it's going to affect the membrane potential. Okay? So that's how you work it out. So this is going to move in, you're carrying a 1 positive, negative charge, it's going to hyperpolarize. And hyperpolarizing will be an inhibiting kind of potential because it's moving it further and further from threshold. Because only when you reach threshold, you can have an action potential. Okay? So graded potentials, by definition, these are small sub-threshold potentials. Right? Because if it reaches threshold, what happens? Action potential. And then that's a large change. So these are sub-threshold by definition, and characteristics, they are conducted in a decremental fashion. They go down, okay? And they can either depolarize or hyperpolarize the membrane, okay? So that's it. Those are the types, right? And it's graded. It's one of the characteristics is that the strength of the stimulus is directly proportional to the size of the graded potential. Okay. All right, so that's that. All right, very good. So action potentials, these are large changes, right, in the membrane potential that are propagated along the rest of the cell membrane or along the axon, right? So that's the definition. So in action potential, there's 3 phases, right? So in the exam, I want you to kind of draw this for yourself. Okay, this is resting. Then we have an action potential, and then we have a thing like that, and then it does that. Okay, so you write for yourself. This is Na plus in. Okay, making it more positive, right? So this is 0, and this is plus 30. Okay, for each specific tissue, the action potential always goes to that same value, whether it's plus 30 or plus 20 or whatever. It doesn't go more than that. For each tissue, it's got a specific. So that means it's all or none. Okay, it either happens and it goes to the max, or it doesn't. Okay, so this will be depolarization. So I'm just going to say dip, and on this side, it's K plus out, right? Because you're now going back to negative, so you're taking the positive charge out of the cell. Okay, so that's repolarization. So I'm just going to call it rep. Okay, and then there's because this is happening lightning fast, it's going to dip a little below resting, and that's the after potential, sometimes called after hyperpolarization, or you can simply call it hyperpolarization. Okay, because it's becoming more polarized than -70. Okay, and that okay, from here to there is the hyperpolarization. And then the sodium potassium pump kicks in to restore the conditions at rest. And we'll start to pump 3 out, because we just dumped a bunch of sodium in there, and then potassium in, 2 in, okay? Because the potassium is kept outside to remove the positive charge. Okay? So that's the action potential. Draw that for yourself and write which ion is doing what and which process it is. So we get depolarization, sodium, repolarization, potassium. After hyperpolarization, it goes further than -70. So it hyperpolarized. Okay, so the characteristics of an action potential, I covered some of them when I was talking about that. It's all or none. Okay, it's a large membrane potential change, right? Okay, and it includes these 3 phases where sodium rushes into the cell, depolarization, potassium rushes out at maximum potential, and it repolarizes, going back to polarization. And then there's also hyperpolarization or after potential. Okay, so all those, and there's another table again that shows all the characteristics of these action potentials. Okay, so you go there, you look at that. Okay, so the action potential is got 2 refractory. There's 2 kinds of refractory period. Okay, the first one is absolute refractory period. It goes all the way from here to there. Okay, this is absolute. Okay, and that means it will not respond to any stimulus regardless of the size of it. Okay, the absolute refractory period is what ensures unidirectional flow of this action potential. Okay, because when an action potential happens on a particular part of the membrane, it will generate a current that's sent forward, generate a current that's sent backwards, because the current will just go around it. Okay, so if this part of the membrane going through the action potential is in absolute refractory period, this current is not going to be able to generate anything here. It will only be able to generate current in front of it. Okay, when nothing is happening, so that the action potential only goes in one direction. Okay. Sometimes I might ask a bonus and say, okay, what is responsible for ensuring that action potential is traveling in one direction? Then you talk about absolute refractory period, because at that point it will not respond to anything. Okay, and then the part that is in relative refractory period, which is in front of the action potential, is what can be stimulated. And so the action potential only goes in one direction. So the part where it hyperpolarizes, okay, from there to there, that's relative. Because the strong action threshold potential can start an action potential. Okay, and parts further down will be resting so they can be stimulated. So the action potential only goes in one direction. Okay, so be able to tell me about absolute refractory period, about relative refractory period. Okay, so since I just talked about how the action potential only goes up to +30 for a specific tissue or +20, whatever it is, it doesn't go more than that. So how can we tell how strong the signal is when the action potential only goes to +10? It's the frequency. Okay, the frequency at which you generate these action potentials talks about the strength of the stimulus. Okay, so if you get a sub-threshold stimulus, okay, so that's a weak stimulus, right? It doesn't reach threshold even. It's just a graded potential that in decremental fashion will fade away. So nothing happens. Okay, so 0. And then over here you get a sub-maximum. So that means it has gone to threshold, but it hasn't quite gone to, to all the way to max. Yeah, you might get like maybe 3 APs, right? And if it goes or more, or whatever, it's relative. And then here, maximal or supra-maximal, you get like 20 or something, I don't know, action potentials. So that's maximum. So this is the strongest signal you can generate. If it goes to supra-maximal, it can only generate that many action potentials per unit time. So it will generate 20. And if it goes way over 20 still, it doesn't say, oh, because I'm slightly more than maximum, so I'm going to go 23. No, it just goes to that. Okay? All right. Anything below maximum is what kind of generates a little bit more or less. But once you reach max, that's it. It doesn't matter how max you go. Okay? So the frequency of the action potentials will determine the strength of the signal, which is the frequency is simply the number of action potentials that are being fired per unit time. Okay, like per second or whatever. Okay, so propagation of action potentials, they travel in one direction because of the absolute refractory period. All right, and then myelination, we just talked about it, speeds up transmission because the impulse is only transmitted in the nodes of Ranvier, which are the spaces between. Okay, which gives that saltatory conduction. Conduction, which means jumping. The myelin sheath is only being propagated in the nodes of Ranvier. Okay, so those are the action potentials. Okay, so we're done with that. Now that we know how the message is conducted, now we want to see how the message is passed on to the next step. So we talk about events at the synapse, which can be between 2 neurons or between a neuron and a muscle cell, which we did when we did the neuromuscular junction, right? Okay, so synapse. So synapse, what did we say by definition it is? What's a synapse? It's a functional connection, right? It's not a physical connection between either the neuron and the muscle cell or the neuron and another neuron. So, okay, it is the synapse is kind of like 3 anatomical structures, okay, such as the synaptic one terminal, right? That terminal, that is the vesicles inside, right? So synaptic terminal, and then after the synaptic terminal, what comes next? It's the gap, right? The gap between the two, which we call synaptic cleft, right? Synaptic cleft. And then after the cleft will be the postsynaptic membrane, which when we did muscles we called it motor end plate, right? Motor for muscle, motor end plate, but simply the postsynaptic membrane. And what's on it? What's on the postsynaptic membrane? What was on the motor end plate? What's that? What did you say? Yeah, ion channels, ligand-gated ion channels. Sodium-gated, like when we did the ligand-gated sodium channels, yeah, ion channels, you're right. What else is it? So, it's got ion channels, okay? Yes, enzymes that break down the neurotransmitter. So, that's the significance of this postsynaptic. The specialized part of the membrane that is at the synapse that has all these receptors for the neurotransmitter, right? You gotta have the receptors for the neurotransmitter for the presynaptic neuron for it to be able to influence what's happening in the postsynaptic cell, right? So enzymes like acetylcholinesterase, etc. All right. So those are the characteristics of it. Synapse, and we get two kinds of synapses, right? The first one is electrical, and electrical synapse occurs in cells that are connected by gap junctions. Okay, and those gap junctions have specialized proteins called connections, right? Okay. Okay. So gap junctions are mostly found in. This is my thing for gap junction. Okay. They are found in smooth muscle, right? And what do you find in cardiac muscle? Intercalated discs. Intercalated discs are just glorified gap junctions. Okay, they are special because they're found in the heart. The heart never stops. Okay, intercalated discs. Okay, and because these cells are connected by these ion channels, which are gap junctions in smooth muscle, intercalated discs in cardiac muscle cells. Okay, whenever you have one of the cells is excited, the membrane potential passes through all these cells as if it's the same membrane, okay, because of these gap junctions. Okay, so that's the function of electrical synapses. It gives the ability of a smooth muscle to contract as a unit. All the muscles contracting as one. Okay, because the gap junctions allow that quick movement of the action potential across all the cell membranes as if it's just one membrane. Okay, but more of it we have chemical synapses, okay, that are responding to a chemical. Okay, where the first cell, the presynaptic cell or neuron, sends a neurotransmitter that will attach on those ligand-gated channels on the postsynaptic membrane. Right? Okay, so these are actually the parts of a chemical synapse. Okay, so I jumped real quick, but all those parts, synaptic terminal, synaptic cleft, because we have talked about it most, is the chemical synapse. Okay, so the electrical synapse we just talked about, those are the gap junctions and stuff. So those are the parts of a chemical synapse, right? Because it's one that sends the ligand to attach on those ligand-gated channels, right? And then activity at the synapse, what can happen in the synapse? You can do two things, right? What can you do? One of them is you can excite it. Excite it to the postsynaptic potential. You can, depolarize the membrane. So you can you can excite it or stimulate it, and then the other part you can IPSP, inhibitory postsynaptic potential. Okay, inhibitory postsynaptic potential. You can inhibit it. Okay, and we've already said one way to stimulate is to open what channels? Sodium channels, because that's the only way to depolarize. And we said IPSP, there's two ways, and we talked about it. We said you can open chloride ion channels, or you can open K+ ion channels, potassium. Okay? Then you hyperpolarize, and here you depolarize. Hyperpolarize. Okay? So now you know all these things. You gotta know these things. I ask about it sometimes. Okay, so there are two ways by which you can stop this neurotransmitter, right? Because we don't have a physical connection, we can kill the messenger, right? So we can enzymatically break it down. That's one way of doing it. Or we can take it back up into the synaptic cleft, which is reuptake. Okay, so if I ask in the exam, what are the ways that you can stop a neurotransmitter in the synapse? We talk about that. Enzymatic breakdown and reuptake, okay? So by endocytosis. So we talked about neuromodulators, right? And we said, so this is our little neuron, right? Presynaptic neuron, and then this is the second neuron, right? Okay, postsynaptic neuron. Okay, so what is modulation? What is the definition of modulators? These are chemical messengers that can presynaptically or postsynaptically affect events at the synapse, right? So a neuromodulator neuron will come and boom, synapse right here. Or it will come boom and synapse right there. Okay? Synapse right there. Okay. So presynaptic modulation, postsynaptic modulation. So this can influence what happens here. Okay? if the message is already sent, this can be either excited further or inhibited. So you modulate the signal, okay? Whether you want to increase it or decrease it, whatever you want. So neuromodulators, they will affect what happens in the synapse, presynaptically or postsynaptically. All right? Okay, so I gave you that table with all those different neurotransmitters and all those conditions, right? And I said I want you to know three. Those are the three, right? So the first one we talked about is the acetylcholine, where you have those antibodies attacking those, you know, on the postsynaptic membrane. Those are acetylcholine channels, okay, ion channels. Okay, so then you cannot stimulate the muscles. So this is a neuromuscular junction, and you get this weakness, and you can't move myasthenia gravis. Okay, so I want you to talk about the neurotransmitter that's involved and what kind it is. Because this one is stimulatory, right? Because it stimulates muscles to contract. So if you are knocking off that ligand-gated channel on the postsynaptic membrane, on the motor end place in this case, because the motor end plate is on the muscle cell, okay, then it won't be able to contract. So your muscles are weak and you can myasthenia gravis. You gotta be able to explain to me how the condition arrives and you know, and the neurotransmitter involved, what kind it is. Okay, this one, serotonin, is inhibitory. It's mostly inhibitory. I chose this specifically because it gives you an example of the three kinds that you can find. Ones that are stimulatory, ones that are inhibitory, and dopamine that is both stimulatory and inhibitory. Okay, so this is inhibitory. Okay, and it inhibits some of this excess noise that happens in your head that can cause depression. Okay, so serotonin, if it's lacking, then you become depressed, right? So what do? You have those serotonin, selective serotonin reuptake inhibitors, those SSRIs. Okay, so they'll block the symport for serotonin, so that it's not taken back up into the presynaptic cell. And then you have more serotonin in the cleft, then you are not depressed. Okay, but these ones, you quickly adapt to them quickly, and they're Zoloft and Prozac and all that. They are selective serotonin reuptake inhibitors. Okay, and so that's that. And then dopamine, I said Parkinson's disease. The inhibitory nature of dopamine is what we are interested in for Parkinson's disease, because it cancels off unnecessary motor activity. Can you drop the phone? Oh, sorry. So it cancels off unnecessary motor activity. So if there's not enough inhibitory dopamine, you have the shakes, right? Okay, and especially intention tremors when you're trying to do something. Otherwise, if there's enough inhibitory dopamine, all that unnecessary motor activity is cancelled off. Okay, so you talk about that. So I can ask about the clinical examples of synaptic function, and then we talk about this disease. Okay, good. So these are the two. We're going to talk about it already. We said this stimulates, excitatory, it depolarizes, involves sodium. Mode of action is depolarization, and then the sodium moves in. Okay, and then IPSP, we said it inhibits inhibitory postsynaptic potential. Sometimes I ask about these two things that can happen in the synapse, and then of course mode of action, Cl- in open chloride channels, or it can open K+ channels out, right? So those are the two scenarios that inhibits in hyperpolarization. Hyperpolarization for short. Okay, good. So gotta know that. Okay, those two events we've already kind of done them. Okay, so chemical synapse activity. All right, those gradient potentials we talked about it. They can be summed up. So there's two ways you can sum them up, right? What's the other? What's one way? It's spatial summation. Spatial summation means you have two or more neurons firing simultaneously, okay? If they're not firing simultaneously, by the time the second one fires, maybe the gradient potential has already disappeared, so it doesn't sum up. So if you are defining spatial summation, you have to say two or more neurons firing at the same time or simultaneously. Okay, and then they can be summated to reach threshold. Then you have an action potential. Okay, so two or more neurons fire simultaneously at the same time. Okay? That's spatial. Temporal. What is temporal? You've got one neuron doing what? Temporal, you've got one neuron. Oh, this is one. It fires two or more action potentials in close succession of each other, right? So, two or more APs, two or more action potentials close to each other. Okay? It's important because why? When one hits and it starts to depolarize or hyperpolarize, the other one will hit while it's still hyperpolarizing or depolarizing, and then it ends up. But if one comes and then the other one comes tomorrow, the gradient potential is gone. Okay, so they have to be one neuron firing two action potentials close to each other in close succession. Okay, so that when one hits before the gradient potential goes, the other one hits, and that's how you summate. Okay, two kinds of summation. Usually I ask about this in the exam, and can you define for me spatial summation and temporal summation? Okay. All right. So in real life, the way these axons, these neurons are all connected to each other, is such that sometimes you can have one that is bombarded by many. Okay. So you have many converging onto one. So you have many going to few, right? So you have 10 or so, maybe going to 1 or 2, okay? Neurons, they're converging onto one, to converge, to convene, to come together. So you have many, so that you have many parts of the nervous system that can bring different kinds of information that can then be processed by one or a few neurons, so you can generate a response that is coordinated. You know what I'm saying? So that's the integration part of the brain. Because if something is happening here, and then something is happening here, and they're all independent, the brain cannot put it together and deal with it as one. Because the brain has to give one response. So that's what convergence allows. And then a few to many. To diverge is to go out, like this. So you may have like 2 or 1 going on to 10 or whatever, okay? So it will diverge. So one neuron can send a message to different parts of the brain so that they all respond in unison together. See what I'm saying? So that's the function of divergence and the function of convergence I just talked about. And then we talked about how sometimes we have this arrangement here. So this is the presynaptic neuron, right, in the terminals. And then we have the postsynaptic neuron here. Okay, so this one we said it has branches called collateral branches at right angles. It will come back and synapse with that. Okay? So it forms like a circle, right? So that's oscillating, right? So when the postsynaptic neuron collateral branches come back and synapse with the presynaptic neuron. So that's oscillating. Sometimes I ask you to define these. They're called neuronal pools, neuronal pathways, and circuits. Okay? Very good. Now we're done. So we go to the endocrine system, right? So in the first one, we were really talking about just the general functions, the general receptors, and and the kinds of hormones they are. Okay, and then we kind of sort of talked more about water-soluble hormones, and then the second one we just focused on liquid-soluble hormones and the amount of action. Okay, so as usual, we always talk about the functions of each system that we talk about, right? What is metabolism? How is the endocrine system involved in there? Okay, things like T3, T4, the thyroid hormones, they affect metabolism, mobilization of glucose, all right? Even cortisol, which we said is also a stress hormone because it mobilizes glucose. Whenever the body is stressed, the natural response is to give nutrients. That's why when people say I'm doing comfort eating when they're stressed, because the natural survival instinct is to get nutrients. So you go and eat and you feel better. Okay? Okay, but the problem is that you don't understand. Okay, so ion regulation, things like aldosterone, okay, that regulates sodium, so you retain more sodium. If you release aldosterone, okay, you retain more sodium, and then you retain more water with it. Wherever the ions go in the kidney, water follows it to dilute. Okay, so if you want to increase your blood volume, you retain more of these ions, and then water follows. Then you release concentrated, low volume urine. If you want to get rid of fluids, let's say you got high blood pressure, then you release more ions out, okay, and then water follows it, so you have more dilute urine, high volume, okay. So, and also mineralocorticoids. Remember those mineralocorticoids that deal with minerals, okay, of which dosaline is one, or mineralocorticoids. So all that is ion regulation. So you can talk about that. Give me examples. You know what I'm saying? Okay. Water balance. When you talk about this, things like ADH, antidiuretic hormone, because you retain more water by retaining more ions, more sodium, more water is retained, and then you have concentrated in blood. Okay, so water balance does that, and then immune system, the thymus, that's what thymosin. What are thymosin and thymopoietin for? What are they for? Maturation of what? T cells. That's the involvement. Because in the exam, if I ask about the functions of the endocrine system, I want you to talk about all these things and give me some information about it. Give me some examples and say, okay, this is what's happening, and this is why the endocrine system is involved in immunity. Okay, so heart rate, blood pressure, again, it's all related to ion regulation and water balance. Specifically, when the heart is, like the atria of the heart are being stretched too much because a lot of blood is going back, because you've got high blood pressure, you've got high blood volume, the right atrium will release atrial natriuretic factor or hormone. Okay, and this goes to the kidneys. It will cause the kidneys to excrete more sodium. Natriuretic means uresis is anything that's being lost in urine. Okay, nat is sodium. That's why Na is the symbol for sodium, right? Okay, so atrial natriuretic peptide will cause the release of sodium in urine, and water follows it. So you release more dilute, copious volumes of urine, and blood volume goes down, blood pressure goes down, and you're good. Okay, so that's how it's involved in blood pressure and heart rate. Okay. All right. So, what else we got here? Glucose and other nutrients. When you eat a meal, what happens? What hormone do you release? Insulin. Okay. If you're diabetic, you're prone to that. Okay. If you don't eat a meal and you've got low blood glucose, what hormone do you release? Glucagon. And what does glucagon do? It goes to the liver, breaks down glycogen into glucose. Okay, so you talk about all that, all right? And then, of course, reproduction. This is the obvious one. Things like those androgens, testosterone, and estrogen for females. Okay, one causes spermatogenesis, the other one causes oogenesis for egg cell formation. So, testosterone, estrogen, all those androgens involved. And then, of course, uterine contractions and mammary gland contractions. Okay, oxytocin does that. All right. All these are hormones. Okay, oxytocin is released by the posterior pituitary when we are done. So, talk about these functions and give me some detail. Okay. Okay, so we talked about it. We said the endocrine system is the second largest control system after only the nervous system. Right? Okay, so and we said there's about 3 of those glands, endocrine glands in the head: the pituitary, hypothalamus, and pineal body, right? And then we said the rest of them are located all over the body. You know, with the thyroid, the thymus, the pancreas, the adrenal glands, all those. Give examples of all those, and what else? And then there are also other parts like the GIT, the duodenum, and so forth. The liver produces, the kidneys produces hormones, and so forth and so forth. But the glands are those other ones that we talk about, thyroid, thymus, adrenal glands. All right? Okay, so you mentioned that if I ever ask about, some of these hormones. So you always have to compare these two systems, right? Because we said the one is the first and the other is the second control system. So this is a good thing to compare these two. It's a good question. So we know the organs of the central, of the nervous system, right? We just did them. We said the brain and spinal cord, the nerves and so forth, right? And then the organs of that, we just talked about the thyroid, thymus, or those ones that produce glands. So you talk about that when you compare those two cells. Cells that produce the hormones, all right? The cells of the glands and so forth, and then the cells of the nervous system. We just talked about them, the two neurons and glial cells. So this would be a nice question to do, right? If I ever ask this, just talk about these things. The target cells for the endocrine system. Any cell that is a receptor for the hormone is a target cell, right? And then for the nervous system, wherever there's a synapse, functional connection, okay? When one cell synapses with another cell, or muscle cell, or neuron, then that's how it affects the other cell, okay? When there's a synapse, all right? Communication, how our hormones are passed along in the body. Hormones travel in blood, all right? And then we know about the nervous system, right? How does that happen? Via action potentials on the axon, right? That's how it does. So this question will be something that you know, and you just compare those two systems and no problem, right? Speed of transmission or speed of activity, the nervous system is fast, right? Step on a tap, like I always say, before you know it, it's in your brain already. You feel it and you retract your leg. Okay, so that's lightning fast, right? Milliseconds time. But on the other hand, the endocrine system is system-wide, and it is longer duration, especially for lipid steroids, right? Because those ones stay much longer, and they got a longer leg phase. You got to make a protein first before you feel the effects of that. So the endocrine system, on the most part, is longer and slower. The nervous system is faster and whatever. Okay, right? So it's shorter. And then, of course, modulation. We said action potentials are the same. They never go bigger or lower. So how do we determine the strength of the stimulus? By the frequency. So nervous system is frequency modulated. How about the endocrine system? How is it modulated? How do we know this is a very strong stimulus? What's that? Amplitude, yes. That's the concentration of the hormone. If you produce a tiny bit of hormone like this, that's a weak signal. But if you produce a copious amount of that hormone, strong signal, guys. Okay, so one is amplitude modulated, which is concentration of hormone, amplitude, and the other one is frequency. We just did the nervous system right now, okay? So talk about all those differences, and this will be a nice question to do actually. I'll probably ask it in the exam, 5 points, okay? Maybe give you 2 or so bonus points, and then you talk about other differences, okay? So hormones, of course, these are chemicals that are released. Where are hormones released? Into the interstitial fluid, right? And then they diffuse into blood. So that's how you define hormones, right? They are either proteins, water soluble, or lipid soluble. Lipids, which are lipid soluble, okay? And so these ones are water soluble, H2O, and these ones are lipid soluble, okay? And therefore these ones will have those transmembrane proteins because the membrane is lipid and it can't go through it, okay? And then this one will have either an intracellular, either a cytoplasmic or a nuclear receptor because it can go inside, okay? So how do you control the secretion of these hormones? Okay? For starters, you could use other compounds, other non-hormone substances like glucose in the blood affects the release of insulin. Okay? And then also there are times when the nervous system can stimulate the glands to release. Okay? Such as how the hypothalamus controls the posterior pituitary, right? That's via nervous stimulation, action potentials. Or you can have other hormones affect the release of other hormones, like how the hypothalamus releases growth hormone, releasing hormone, so that the anterior pituitary releases growth hormone. Okay? Okay, so you talk about that. So if I ask about secretion of, how do you control the secretion of hormones, you talk about those three things. Okay, how do you control secretion of hormones? That. So water soluble hormones, okay, these are soluble in water, and so they're charged particles, right? And therefore they are easily transported in blood, right? Because blood is an aqueous environment, it's water. Okay, and then of course soluble, they're only soluble in lipids, okay, and therefore they are transported bound to a protein, okay, to make it soluble, to make it exist in an aqueous environment, okay. So we talk about those things. So and then if I ask you, how are hormones excreted from blood to come out of blood, pretty much. So one of the ways is that when it reaches the target tissue, it's going to exit the blood and get into the cells. So that's one way of getting rid of the hormone, right? From blood. Okay? Or you can have the kidneys. They can excrete it. They can excrete those hormones. That's one way of getting rid of the hormone. Okay? And the liver can do that too. Via the digestive system, you can get rid of it. Okay? So I can ask about ways in which hormones are the control of secretion. Okay, those three things we just talked about. And then the control of excretion. Those are two different things. So beware of that, because I might just ask one or both. Okay, so 2-point question, with the freebie bonus for the other one. Okay, excretion is those things. When it enters target tissue, when the kidneys can excrete and the liver does. Okay. All right, so very good. So now we have classes of receptors. So we have the membrane bound. So these are bound to the membrane, and these are transmembrane proteins. So that means they span the width of the membrane, right? So these are the lipid bilayer. So it will span the width of the membrane, because then the protein-soluble hormone doesn't have to cross the lipid here. It will just attach out here, and then it will affect what happens inside the cell. Okay? So that's what happens. That's the mode of action. The hormone attaches on the outside, okay, and then it will cause those G proteins. Remember those G proteins to bind when it changes shape. The G protein binds with this alpha, gamma, and beta subunit. All right, and then once that happens, the GTP on the alpha subunit is substituted for GTP. Remember those three steps I talked about. And then once that happens, the G protein will dissociate from the receptor. Then the alpha subunit attached to the GTP will detach from the gamma and beta subunits. Okay. It is the activated alpha subunit that will then go and attach to, say, adenylate cyclase and cause the production of cyclic AMP, or it can phosphorylate other enzymes, okay, and so forth, right? Or it can attach to an ion channel. And then the ion channel opens up, and then calcium gets in, for example, calcium. They attach to commodity, and that's how the smooth muscles contract. Okay? So you can do several things with this. So that's mode of action. Sometimes they ask about that, whether it relates to ion channels or activation of another enzyme, because usually it activates protein kinases, which then phosphorylate other enzymes, and then activates them. Okay? So the intracellular messages, intracellular mediators, are also called second messages, like cyclic AMP, cyclic GFP, guanine monophosphate, okay, all those ones, okay. And then, like I said, they can also phosphorylate other intracellular enzymes, and when they are phosphorylated, they become active. They can do that, okay. So, gotta know the mode of action of these water-soluble hormones, of course, which is the G proteins, right? And then, of course, intracellular receptors. This is specifically for lipid-soluble hormones. They can either attach to the cytoplasmic receptor. This will be the very, very non-polar ones. Okay, so they really hate water, so they have to be attached immediately to another protein as soon as they exit blood. So they attach to a cytoplasmic receptor, and the other ones may be a little bit polar, so they can kind of survive the aqueous cytoplasm environment, and they can go straight to the nucleus and attach to the nucleus. Okay, all right. So that's that. So we move on to the last one, where we're pretty much talking more about lipid-soluble hormones and their mode of action. Okay, we said these ones are attached to intracellular proteins. I was just talking about those intracellular receptors, which are proteins here. or a nuclear or a cytoplasmic receptor. Okay, so it's the hormone receptor complex that goes into the nucleus, attaches to those hormone response elements on the DNA, and causes the transcription of a specific gene. A specific gene is just a code for a protein chain, okay, for one protein. And then you create messenger RNA during transcription. It goes out, messenger RNA into the cytoplasm, and it attaches where? Free ribosome. And then what happens there? Translation. You translate that transcribed message into a protein, a sequence of amino acids. And then the effect of that protein will be the effect of the hormone, right? So you gotta know that. So these are slow acting because you gotta do all that. Produce messenger RNA. After that, you gotta produce the protein, and then after that, then it goes into blood and it does this thing. Okay. So the mechanism of action, I just talked about it, right? Attaches to a cytoplasmic or nuclear receptor, and it causes protein synthesis. And you all know about protein synthesis, right? What it involves, creation of messenger RNA. So now we go to how the hormone, the hypothalamus controls the pituitary. So the posterior pituitary at the back is what is called the neurohypophysis. It's controlled by action potentials, all right, from the hypothalamus, all right. And the anterior pituitary is the adenohypophysis. It's controlled by the hormones that are released by the hypothalamus. For example, growth hormone, releasing hormone, which is my favorite. And then the other one, and it causes the anterior pituitary to produce growth hormone. Okay, so because this is an action potential, it's called the hypothalamo-hypophyseal tract. From the hypothalamus going to the hypophysis, which is the in the tract we know about. The tracts are axons in the central nervous system, right? So it's a tract, and then in the outside it's called a nerve. So there's nothing very difficult about this. It's just a long word, that's all. And then this one here, the hypothalamo-hypophyseal portal system, because it's hormones, and where did we say hormones travel? In blood. So there are blood vessels that carry the hormone from the hypothalamus into the pituitary. So it's a portal system, that's all. Portal just means carry your blood vessels from one organ to the next organ. That's all. Nothing fancy. Okay, so you gotta be able to tell me. Sometimes I ask about how is the pituitary, the posterior and the anterior pituitary controlled by the hypothalamus. Talk about that. 2 points. Okay, so the thyroid gland is the one that has the characteristics. It's got all those lateral lobes, right? It's got a pyramidal lobe up here, the isthmus that connects the two, okay? And then it's got the follicles. And inside the follicles, those are those little globular sort of bags in there that contain thyroglobulin, right? The thyroglobulin is there because T3 and T4, tetraiodothyronine and triiodothyronine, are lipid hormones. So they need to be attached to a protein. Okay, so that's what the thyroglobulin does. So that produces the T4 and T3. The parafollicular cells, which are the cells surrounding that, will produce the calcitonin, which we've seen before, and we know what it does because we did it with the skeletal system. All right, so that's what it does. So T3 and T4, you gotta know the effects of T3 and T4. Remember when we were talking about how it increases metabolism, by mobilizing glucose, and how it affects maturation and reproduction and growth of tissues. Okay, so all those effects, okay, gotta know. Right, so, and you can look them up, okay? So, sometimes I ask what are the effects of thyroid hormones? T3, T4, thyroxine, which is tetraiodothyronine. And then, of course, at the back, there's 4 dots called the parathyroid gland at the back of the head. We're probably going to take 5, just like we did in the morning. So, they produce parathyroid hormone, which we know all too well about, right? Remember, we did it in the skeletal system. It was even in the exam, okay? And it produces active vitamin D for absorption in the small intestine of calcium, okay? It can also break down calcium from bone, activates osteoclasts, right? We talked about that. Or it will cause more reabsorption of calcium in the kidneys. Okay, so we all know too well about this. If I ask about this, it will be like a free before you. You get 3 points for nothing. Because we've done it before, okay? So we all know about that, so I'm going to go quickly on that. Now this one here is got the adrenal glands, it's really like 4 glands in one. Why? Because it's got the adrenal medulla in the middle that produces epinephrine and norepinephrine, right? Fight, flight stuff, right? And therefore, you gotta know the effects of that, like increased blood pressure, increased heart rate, increased glucose to muscles, and so forth and so forth. Sometimes I ask, what are the effects of, adrenal medulla hormones? And you're like, which one? It's epinephrine, okay? Okay, so, or sometimes I may say, what are the effects of epinephrine? Depends on what mood I am, okay? So, now the adrenal cortex is the outer layer. It's split into 3 layers, and each one produces a different kinds of hormone. Okay, so zona glomerulosa is one that does all those mineralocorticoids, right? That deals with minerals. And zona fasciculata, glucocorticoids like cortisol, okay, for glucose mobilization and so forth. And then zona reticularis, there's gonadocorticoids, which is androgens. It produces androgens, which are the precursors for testosterone and estrogen. Okay, so it's like 4 glands in one. Okay, all in the adrenal gland. Okay, and then the pancreas, we said the exocrine function produces digestive enzyme. We're not interested in that right now. Okay, we do it when we do the digestive system, but we're interested in its endocrine function. Okay, where we have the pancreas, pancreatic islets, or islets of Langerhans, sometimes called, with the beta cells that produce insulin and the alpha cells that produce glucagon. Okay, so you gotta know that. Sometimes I ask about the source of these hormones. And the pineal body really doesn't have characteristics that are well known, functions per se, but it does sort of has some bearing on reducing reproduction and so forth. But the big function of it is to produce melatonin, which is involved in sleep-wake cycles. All right. So that's that. So that's the pineal body. The pineal gland or pineal body is one of the ones in the head, right? It's in the head. Okay. So the thymus, we talked about it. It's involved in immunity, and it produces thymosin and thymopoietin, which are involved in T cell maturation. Okay. So, and the thymus, of course, is very big in the infants, and it gets smaller in adults. Okay, because they are now exposed and so forth. Okay, and it's got all those lobule as well. It's covered by a capsule. Okay, so there are also other organs in other systems that happen to have cells that produce hormones. Okay, like in the small intestine, you've got, of course, the stomach that produces gastrin. It's a hormone that goes in blood and causes gastric secretions. And then you've got the small intestine that produces secretin and cholecystokinin. Secretin causes the production of alkaline fluid from the pancreas, okay, so that you neutralize acid from the stomach, okay, and then cholecystokinin is involved in fat digestion, okay, so it will bring down bile from the gallbladder for fat digestion. So secretin, cholecystokinin, just so you know, erythropoietin, we talked about it last time, it causes the production of red blood cells, erythropoietin from the kidneys, and renin, which is involved in the the blood pressure, because it converts angiotensinogen to angiotensin 1, right? Which is then goes to angiotensin, which is involved in increasing blood pressure. That's why you have ACE inhibitors, angiotensin converting enzyme inhibitors for blood pressure medication, okay, because of that. But kidneys produce renin, and of course erythropoietin for red blood cells. And then of course the placenta will produce human chorionic gonadotropin, which maintains the corpus luteum and maintains pregnancy, because the corpus luteum will then produce estrogen and progesterone, which maintains the lining of the uterus. So HCG, and also the placenta will take over the production of progesterone and all those hormones and estrogens. Okay? So just for you to know, the last thing I want you to know is these autocrine and paracrine substances. These are hormone-like substances, but they're not quite, okay? So the autocrine ones will affect similar cells locally, though, in the same area, in the immediate vicinity, okay? And then paracrine ones will affect different cells, different cell types. Like growth factors will affect different kinds of cells, because everything grows, right? Not just one type of cell grows. So it will affect, but in the same area. Then the last thing I want you to be able to do is to be able to compare the hormone-like substances and actual hormones. Okay, hormones are produced by specific glands, whereas the hormone-like substances just come from the cells surrounding. So there is nothing specific. And sometimes the functions of hormone-like substances are not well known. Okay, they're not very clear, like the specific functions of hormones. Okay, and the very last thing, they only have local effects. Hormones are released into blood, they're systemic, it goes system-wide. Okay, so that's it. Thank you, ladies and gentlemen. That's it. I took about 5 or so, yeah, a little bit more, but I can give you some. We can meet at 5. Right? Thank you, guys. I appreciate it.